All right, so good morning. So this is AMP1. So we just want to go over a few things before we get started. So again, here's where you access MyLab and Mastering. If you registered your access code, we'll say Open MyLab. Go to your home page. It may not look exactly like this. This is mine, but you can get to your different assignments there. Here's where you get to your e-text. You click that. It'll take you to the e-text, and so you can read the text online. You can change the font, go to different sections or different chapters. You can create bookmarks. Okay, you can take notes here. You also have access to a study area. You can organize it by chapter. You can look at different resources. Study by chapter. So chapter 2, you got practice tests, pre-tests, matching, multiple choice, true, false, fill in the blank, essay quiz, the different sections of the e-text, glossary, flashcards, some animations, and MP3 to listen to. So you have those resources in addition to what's in Canvas. All right, let's see. All right, so for chapter 2, remember I have the learning activities. So you got lots of notes, lots of videos, links to the book, links to those activities from the study area. Here are your assignments for week 3, chapter 3, notes. You got some videos to watch. Okay. These playlists, I think, are going to be really helpful for you for chapter 3. I believe you had one for chapter 2 as well. Yeah, YouTube playlist, chemistry, so make sure you look at that. All right. We got some different games to help you learn the organelles. Okay. All right, more animations. Let's see. All right, so we'll also pull up the review sheets. All right, so this one's for exam 2, so we don't really need that one. Go back up to chapter 1. All right, exam 1 review. So for chapter 3, parts about the cell membrane transport, concentrations, organelles, parts of DNA, cellular processes, mitosis, meiosis, transcription, translation, which we'll learn about this week. Prokaryotes versus eukaryotes, concepts of cell theory, some historical people and what they're known for. Okay, that one makes a really good matching question for the exam. Characteristics of different parts of the cell, diffusion, phases of mitosis. You should know at the end of mitosis how many cells you're going to have based on how many you start with. Factors that affect diffusion rates, we'll learn about that. Possible short answer essay question for the test will be to explain transcription or translation. You could also be given some strands of nitrogen bases. We haven't covered this yet, but I'm just introducing the concept to you. You might have to make a corresponding mRNA strand, which we'll learn about. Okay. All right. And then some bonus is not just making mRNA, but also making a coding strand of DNA and tRNA anticodon. Again, you don't know any of this yet. That's what we'll learn in chapter 3. All right, let's see if we can pull this up. So we're still finishing up chapter 2. Okay, this week we've got we're finishing up chemical level organization, starting cellular level organization, finishing up chapter 2, starting chapter 3. Yeah, the Mastering AMP quiz over chapter 2 this week. All right. The exam over chapters 1, 2, and 3 is next week. Okay. Likely sometime on next Thursday. I typically just give you a range of time to finish it. All right, let's see. I also want to make sure you remember down here, the open study room. Make sure you take advantage of that. These are the hours that they're open. You can just go in there and study Monday through Thursday. If you want a tutor for A&P lab and lecture, Tuesdays between 11 and 1 and 2 o'clock to 3:45. Okay, so just make sure that if you want tutoring for that, you can always, contact me, but that's another resource that you have available to you. She does track attendance, and then she reports the attendance back to me at the end of the semester, and I can use that, basically to, someone's on the borderline on a grade, I can use that to determine if they get an extra credit or not. So definitely take advantage of it, because I typically don't do much extra credit. All right, so let's go ahead and see if we can finish up chapter 2. Don't think we got this far. I think we were just getting into properties of water last time. Is that correct? Y'all remember? Okay. So all right. So properties of water, 2/3 of your body weight makes different solutions, which are just mixtures of substances. Any solution is going to have two main parts. It's going to have a solvent, which is your liquid, and your solute, which is what gets dissolved in the solvent. Okay, so solutes get dissolved in the solvent. Okay, water is a universal solvent, so that means many molecules are going to be water soluble. It's very reactive, can be used as a reactant in many chemical reactions. It has a high heat capacity, which means it just takes a lot of heat to raise the temperature of a unit mass of water by 1 degree Celsius. All right, let's see. And lubrication is also used to moisten things and to reduce friction. All right, so pause. Okay, so properties of water again. Water's polar. Remember, it has a positive and a negative end. Hydrogens are more positive. Oxygen is more negative. So many of these inorganic compounds can split into different smaller molecules once they dissociate in water. So things like ions. So ionization is when you take, say, a salt and it separates into ions when you mix it in water. So if you take sodium chloride, mix it in water, sodium ions will separate from the chloride ions. And polar water molecules will form, we'll see a picture of this, what we call hydration spheres. Hydration spheres are basically circles that surround small polar molecules. Okay, it's a bunch of water molecules that surround it. Keep them in solution. All right, so again, this is water being polar because of the negative pull of the oxygen, electrons in the oxygen. You have a negative pole. Hydrogen doesn't pull as strongly on the electrons in the covalent bonds, so it's slightly hydrogen or slightly positive. All right, so here's an example of the hydration spheres. So chlorine is negatively charged, so the positive hydrogens are going to surround the negative chlorine ions, and the sodium ion is positively charged, so the negatively charged oxygens are going to surround the positively charged sodium ions, and it keeps these ions in solution. All right, so here's another example. This is glucose, and it's being surrounded by the water molecules. Okay, it doesn't dissociate like ions do, but it does dissolve in solution. So it's basically water molecules are surrounding the solute. All right, so talking about ionization, remember these are ions that dissociate in solution. They come apart, they separate. Remember, associates come together, dissociates come apart. So sodium chloride in water will become sodium ions and chloride ions. Potassium chloride solution will become potassium ions and chloride ions. Calcium phosphate dissociates into calcium ions and phosphate ions. Sodium bicarbonate dissociates into sodium ions and bicarbonate ions. Magnesium chloride ions separate into magnesium ions and 2 chloride ions. Sodium hydrogen phosphate will dissociate into 2 sodium ions and 1 hydrogen phosphate ion. Sodium sulfate will dissociate into 2 sodium ions and 1 sulfate ion. And you'll see that in different biological situations. All right, electrolytes and body fluids. So you hear that, electrolytes when you drink your Gatorade or Powerade. So what are they? They are inorganic ions that can conduct electricity in your solution. So it's not just that you need to replace them because you lost them because you sweated. You also need them so that your body can actually conduct electricity the way it needs to. Now, you don't want it to conduct electricity in ways that you don't need to, but you do have some electricity that happens in your body. All right, so electrolyte imbalance can disturb your body function. Okay, water is considered to be hydrophilic. I mean, water is obviously going to like itself, but other chemicals that like water, hydro means water, philos means loving. So if they interact with water like ions and polar molecules do, they are hydrophilic. They're hydrophobic. Phobos, same root as the word phobia, which means fear. These are nonpolar molecules like fats and oils, and they do not interact with water. So it's like the greasy dishes that you leave in the sink overnight, the grease rises to the top. Okay, those are hydrophobic molecules. They clump together and separate from the water. Okay, colloids and suspensions. So colloids are solutions that contain dispersed proteins, other large molecules. Blood plasma is a colloid, so it's a kind of a biological application. Suspensions have large molecules and they settle out of solution, and that's your whole blood. So whole blood's a suspension, blood plasma is a colloid. Colloids, they disperse. Suspensions, particles can settle out. Okay, pH and homeostasis. So pH, the formula basically means it's the negative logarithm of the hydrogen ion concentration of a solution in moles per liter. So what does that mean? Well, it basically just means that this logarithm it's a base 10 equation, so it's an exponential equation. So it's basically the more hydrogen ions you have, the lower pH you're going to have. The fewer hydrogen ions you have in the solution, the higher your pH is going to be. Okay? So neutral pH means you have a balance of hydrogen ions and hydroxyl ions. An example of that would be water, which has a pH of 7. Okay, so your pH will be considered acidic if the pH is less than 7. And in that scenario, you have a high concentration of hydrogen ions because, again, it's the negative log of the hydrogen ion concentration. All right, so high concentration of hydrogen ions, low pH. The flip side of that is that the OH concentration is low. Basic solutions or alkaline solutions have a pH that's greater than 0. And if their pH is greater than 0, that means that their hydrogen ion concentration is low. Okay, because again, negative log of the pH, negative log of the hydrogen ion concentration. Okay, so a high pH would mean that the concentration of hydrogen ions is low because that negative in the equation. So that also means the hydroxyl concentration is high. So what's the pH of human blood? Well, it's not quite neutral. It's slightly basic, so 7.35 to 7.45. So just slightly basic. All right, so pH scale. So it has an inverse relationship with the hydrogen ion concentration. Again, that's what we said. More hydrogen ion concentrations, lower pH. Fewer hydrogen ion concentrations, higher pH. Okay. Acids have more hydrogen ion concentrations and a lower pH. Bases have fewer hydrogen ion concentrations and a higher pH. Okay, so here's a pH scale with hydrogen or with acids. So water being neutral, urine is in this range, pH of about, up goes to about 4.5. Saliva and milk are between 6 and 7. Grapes and tomatoes have pH of around close to 4. Beer, vinegar, wine, and pickles have a pH close to 3. Stomach acid has a pH that's close to 0, or close to 1 rather. And hydrochloric acid has a pH of 0, which is as acidic as you can get. Completely dissociates, completely gets rid of its hydrogen ions. Okay, acids, bases, and salts. So this is kind of a continuation of the pH conversation. So acids are proton donors, and protons are just another form of, another way of describing hydrogen ions, because hydrogen ions, remember, let's think back to our discussion of the periodic table and elements and atoms. Anybody remember how many protons hydrogen has? One, very good. So a hydrogen ion is an ion that lost its electron. So if you had one electron and one proton, now all you have is one proton, you're a proton essentially, okay? So we call hydrogen ions protons. So an acid is strong if it dissociates completely in solution. So remember I showed you a table of different chemicals and what do they do when they get in water. If they dissociate completely and get rid of all their hydrogen atoms, or all of their protons, they are a strong acid. If they don't, they're a weak acid. All right, bases are proton acceptors because remember they have a lot of OH concentration. So they'll take those hydrogen ions. They'll remove the hydrogen ions from solution. And so a strong base will also dissociate completely in solution. So acids are proton donors. They get rid of the hydrogen ions. Bases are proton acceptors. They will take the protons, the hydrogen ions. Weak acids, weak bases do not dissociate completely, and they help to balance the pH. So you may think being, weak is bad, but in acids and bases, being weak is good because it helps to balance your pH. You don't want big swings in your pH in your body. Right? You want your pH to stay relatively the same because if it gets too acidic or too basic in your body, your cells can be damaged. Okay, so you want ways to be able to keep the pH in the same range. Okay, salts, these are solutes. Remember, these are things that dissolve in solution. They dissociate into cations. Remember, those are positively charged ions, and anions that are negatively charged ions. And these are other than hydrogen ions and hydroxide ions. So lots of vocabulary words in this sentence basically just means Like we think of salt, when we say salt, we think of sodium chloride. Okay, sodium ions are not hydrogenized, chloride ions are not hydroxide ions. Okay, basically they dissociated something with a positively charged ion and a negatively charged ion. But it doesn't have to be sodium chloride. It could be calcium chloride, it could be magnesium chloride, it could be any combination of a positively charged ion and a negatively charged ion, as long as it's not hydrogen and hydroxide. Okay, we talked about stabilizing pH with weak acids and bases. Buffers are important for that. So buffers stabilize the pH of your solutions in your body, or even just in a chemical setting or in a chemistry lab. So buffer systems will involve a weak acid and its related salts, which would be a weak base. All right, so their role is to neutralize strong acids or strong bases. And they do that, and that helps them stabilize pH. So a classic example of a buffer system is carbonic acid and bicarbonate. So carbonic acid is the weak acid, bicarbonate is the related salts or weak base. Very important in our bodies. Anybody ever have taken antacid? All right. So these use sodium bicarbonate to neutralize the hydrochloric acid in your stomach. You get a little heartburn, right? So that's what they're for. They're called antacid because they're opposing the acid. All right, each macromolecule of life is made up of monomers. And so, okay, we're doing transition here. All right. So, big thing I want you to take out of that last section is pH is a measure of the hydrogen ion concentration, right? Negative log of the hydrogen ion concentration. Lots of hydrogen ion concentration, low pH. Okay? Little hydrogen ion concentration, high pH. Strong acids dissociate completely, get rid of all their hydrogen ions. Strong bases dissociate completely as well. Weak acids do not dissociate completely. Weak bases don't dissociate completely. And buffer systems are a combination of a weak acid, okay, and its corresponding salt or weak base. And we use those to stabilize pH. Okay, so that's really what we want you to get out of that last section. Now, you could be asked to do some sort of calculation of what's the pH. Not necessarily doing a lot of math, but let's just say the concentration of hydrogen ions is 10 to the negative 5 moles per liter. What's the pH? The negative log of that. And so since the log is base 10, and I told you exponent was 10 to the negative 5, basically the log of 10 to the negative 5 is 5. So actually it's negative 5. So the negative times negative 5 is 5. So if I told you the hydrogen ion concentration was 10 to the negative 5, pH is 5. Another way of saying that is what's the difference in concentration between a pH, one solution has a pH of 7 and one has a pH of 8. Well, the one with a pH of 7 has 10 times the amount of hydrogen ions than the one that has a pH of 8. Because remember, lower means more concentration. And since it's log, it's a factor of 10. So I wouldn't necessarily ask you to do real complicated math, but you do need to know each number difference between like 6 and 7, 7, 8, 8, 9 is actually a factor of 10. It's 10 times the concentration. Okay. So it's you look at it like it's just numbers on a number line, but it's actually times 10. So something with a pH of 5 is basically 100 times has 100 times a higher concentration of hydrogen ion concentration than something with a pH of 7. Because it was 2 spaces, 10 to the 2 is 100. Okay, so that's the kind of thing we'd ask you to do. All right, so monomers and polymers. Monomers are the units, and they make polymers. Okay, you add them, you build them together and make polymers. So let's look at examples of that. Okay, so first biomolecule, we're going to talk about our carbohydrates. These are organic because they have carbon and hydrogen. So they also have a lot of oxygen. They use covalent bonds, and they have different functional groups that determine their chemical properties. All right, and I want to be clear, like the section here says carbohydrates, so we're going to go into detail on carbohydrates in just a minute, but this whole page applies to all organic molecules. So I want to make sure that that's clear. So organic molecules could be carbohydrates, lipids, proteins, nucleic acids. Okay. So this is sort of the big picture, organic molecules. Then we're going to focus on carbohydrates, do a few slides, lipids, a few slides, proteins, a few slides, nucleic acids, a few slides. All right, carbohydrates, their carbon hydrogen oxygen ratio is 1:1. You also have a few that are what we call isomers. And you might remember what iso means? Same, good. So they have the same molecular formula, but they're arranged in different structure, okay? But they have the same number of carbon, hydrogen, oxygen atoms. All right, the building blocks, the monomers for carbohydrates are monosaccharides. Everybody say monosaccharide. All right, so those are your building blocks. They're simple sugars made up of 3 to 7 carbon atoms. Examples are galactose, fructose, and I'm sure everybody here has heard of glucose, okay? You may have heard of fructose, like high fructose corn syrup, things like that. That's what fructose is. All right, disaccharides. Okay, so disaccharides, di means 2, so this is 2 saccharides. And so here, this is a process called dehydration synthesis or condensation. So the synthesis part is taking 2 monomers, linking them together. So you synthesize the disaccharide. The dehydration piece is, in order to do that, you have to lose water. So it's dehydration synthesis. Also, because you're losing water, it's called condensation. Examples of disaccharides are sucrose and maltose. Now you also have polysaccharides, and this is where you take many sugars and you condense them through dehydration synthesis. So you're making things like cellulose, starch, glycogen. So it's not just 2, it's a whole long chain, okay? But your disaccharides and your polysaccharides are your polymers, okay? Your monosaccharides, your monomer. All right, so different ways you might see carbohydrates represented. This is called a straight chain form. We just see all the different elements listed out. This is the ring form, and basically what this means is that, without getting too complicated, this ring folds on itself, okay? So what you end up having is you get a 6-membered ring with one of these oxygens being a part of that ring, okay? So this is actually more stable than this one. But you don't necessarily need to know why and all that. We won't get into that. Just know if you see either of these, it's a carbohydrate, okay? All right, and this is a 3-dimensional model because each atom takes up space, so this is kind of what it looks like in 3 dimensions. All right, so formation, here's again the dehydration synthesis. You're taking glucose and fructose. You're linking them together with a covalent bond, but here's an OH and H. You're going to be releasing water. So that dehydration is releasing water, synthesis is creating a bond between these two monomers. So now you have the bond and you release water. So it's dehydration synthesis, also known as condensation. So does everybody understand that dehydration is losing water, like you get dehydrated? Anybody not understand that? Does everybody understand that synthesis is taking two things and putting them together? You're building a new molecule. Okay? So I know sometimes you look at the words and it's like, oh, big words. I want you to make sure you understand what they mean. Now the reverse reaction can happen as well. And here you're using water to break the bond between the two monosaccharides that were connected. So this is a polymer at this point. We're going to break it into two monomers. And that process is called hydrolysis. Hydro means water. Lysis means break. Okay, so it's breaking bonds with water. Okay, so now you're back to glucose and fructose. All right, here's your polysaccharide. This is glycogen. So same process, dehydration synthesis, just over hundreds of times. And you're creating these branched chains of glucose. Okay, this is how your body stores food. It makes glycogen. And then when it needs it to make energy, it just breaks off pieces. All right, lipids. So these are mainly hydrophobic molecules. So again, hydrophobic means they don't like water. Fats, oils, and waxes. Lots of carbon and hydrogen atoms in there. Things like steroids, phospholipids in your cell membrane, glycolipids, glycerides, eicosanoids, and fatty acids. Those are examples of lipids. All right, so your fatty acids, we'll start there. You have carbon hydrogen chains. You'll have what we call a carboxyl end of one of them. So it's of one end of the chain. And it's carboxyl just means COOH. Okay? These are typically going to be non-polar, except for your hydroxyl group. Or sorry, except for your carboxyl group, excuse me. So the carboxyl group has some charge to it, but most of that carbon hydrogen chain is going to be lots of non-polar covalent bonds. All right, fatty acids. Everybody ever heard of saturated fat, unsaturated fat? Okay. So when we say saturated, what we typically mean is it's saturated with hydrogen, right? Usually you say saturated is drenched. So it's like it's drenched with hydrogen, which means there are no double bonds in the hydrocarbon tail. Whereas unsaturated fats do have double bonds in the tail. They can be monounsaturated if they have one double bond, and polyunsaturated if they have multiple double bonds. We'll see an example of what that looks like. All right, so here's your fatty acid chain. Lots of carbon and hydrogen, lots of non-polar covalent bonds, slightly polar here. That's your carboxyl group. But again, don't get too overwhelmed by this. This is just, you see this long tail with the carboxyl group on the end, it's fatty acid. All right, so here's an example of saturated fat versus unsaturated fat. Saturated, and they didn't draw all the hydrogens here, but it basically has all single bonds because it has hydrogen atoms connected to it. Here it's unsaturated because you have less hydrogens connected to these carbons because there's a double bond. So why does that matter? So if the fats are saturated, they can stack on each other, okay? Because they all have the same basic shape and they and stacked where there's basically no space in between. All right, I guess the better way is to like this. But if you think about it in your arteries and your veins, do you want those fats stacking up on each other on the sides of your arteries and veins? No, you don't, because that creates, atherosclerosis and high blood pressure, all kinds of things we don't want. But when you have unsaturated fats, it creates space, harder to pack, and they just basically keep it moving. They don't stay, connected to the side of the blood vessel. And they also don't clump on each other, making it harder for them to get through your blood vessels. So unsaturated fats are very healthy for you. So it's, sometimes we think fat is bad. Not all fat is bad. Saturated fat is not very good for you, but unsaturated fats can be healthy in moderation. All right. So lipids, let's go on to some of these other types of lipids. You have eicosanoids. You have to get them from your diet. You can't actually make these in your body. Some things you can make, your body can make certain chemicals. Eicosanoids you cannot. It's made from a fatty acid called arachidonic acid. Okay, kind of random fact there. Glucotrienes, another class of lipids involved in the immune response. And prostaglandins, these are hormones that have short-chain fatty acids on them. Okay, and hormones are chemical messengers. They cause some sort of response in your body. All right, so here's an example of prostaglandin. So you have basically a 5-membered ring here and 2 tails. All right. One of these tails has a carboxyl group on it, the other does not, but they each have some double bonds in their fatty acid tails. Glycerides, another type of lipid. So glycerides have fatty acids, and they're attached to a molecule called glycerol. And glycerol is basically a 3-carbon compound. It has 3 carbons in it. So it has essentially potentially 3 attachment sites for fatty acids. And anytime you see OL at the end of a word, it's an alcohol. So, most people, when they hear alcohol, they think of ethanol, the alcohol that people drink if they're, over 21, if they choose to. But really anything with a hydroxyl group at the end is a alcohol. And so glycerol is one example. So if you have glycerol with 1 fatty acid attached, it's a monoglyceride. If you have glycerol and 2 fatty acids attached, it's a diglyceride. If you have glycerol and 3 fatty acids attached, it's a triglyceride. So you may also see the term triacylglycerol or neutral fat. It means the same thing as triglyceride. So why are triglycerides important? Well, they provide protection for our bodies, some cushion. They also provide insulation, kind of trap heat inside your body. And they also are a source of energy. All right, so here's your glycerol. Your 3 fatty acid tails, 2 of which are saturated, no double bonds, 1 of which is saturated, does have a double bond. You go through dehydration synthesis, remember you're releasing water, making a polymer. Okay, you get your triglyceride. The reverse reaction is using that water to break these covalent bonds, so that's hydrolysis. Okay. So if you use water to break something, what process is that called? Hydrolysis. If you make a polymer and you release water, what process is that? Dehydration synthesis. Excellent. All right, another class of lipids, you have the steroids. These are carbon structures with 4 rings. So again, this is another one of those that sort of sometimes scares people. If you see the 4 rings, it's a steroid. All right, so don't get too overwhelmed by it. All right, example of a steroid is cholesterol. You have a fair amount of cholesterol in your plasma membranes or cell membranes. Other examples, your sex hormones like estrogen, testosterone, other hormones like corticosteroids and calcitriol. Those are also steroids. And bile salts. Okay. All right. So bile is stored in your gallbladder is released as a bile salt, and then it dissociates and then does its function. All right. So here's our 4-membered ring. Okay. So don't get too caught up in, oh, this is a big fancy molecule with a bunch of stuff going on. 1, 2, 3, 4. Okay. Let's count with me. 1, 2, 3, 4. Okay, so don't get overwhelmed by it. All right, you see the 4 rings? It's a steroid. You might not know which steroid it is. You might not know if it's cholesterol or estrogen or testosterone. To be honest with you, I wouldn't worry about that right now. I would mainly worry about, do you know this is a steroid? Okay, because if you're that far along, you're probably, the answer choices will maybe only have one of these. So I'm probably not going to ask you to tell me the difference between estrogen and testosterone. You might need to know that one is more commonly found in females than males or vice versa, but understanding the differences in the fine details in their structure, I wouldn't worry about that. You just need to know if you see the 4 rings, it's a steroid. Okay, phospholipids and glycolipids. Your cells can make these. So, and a lot of this is kind of how it's, how it's designed. They have a diglyceride, which means 2 glycerides, and there can be attached to a phosphate group. So if it's a lipid attached to a phosphate group, it's a phospholipid. That's what you see in your cell membrane. If it's attached to a sugar, glyco means sugar, like glucose. It's a glycolipid. And sometimes glycolipids are attached to the membrane. All right, generally, both of these structures are going to have a hydrophilic head and a hydrophobic tail. Okay, so heads are hydrophilic, tails are hydrophobic. All right, tails don't like water. And they are structural, which means you see them in the structure of the plasma membrane. Okay. All right, so this is a phospholipid. All right, so this is not a robot, this is a phospholipid. All right, so here's your non-lipid group, your phosphate group, here's your glycerol, the 3 carbons, and your 2 fatty acid tails. All right, so if you see glycerol, 3 carbons, 2 fatty acid chains, a phosphate group, and a non-lipid group, which is basically your head here, polar head. All right, that is a phospholipid. This is a glycolipid. So here's the difference. So this is the same, glycerol is the same, 2 fatty acid chains. Instead of having the phosphate group and the non-lipid group up here, you have carbohydrate, glycolipid. So glycolipid. All right, so another application. So when large numbers of phospholipids and glycolipids are in water, they can form structures what we call micelles. With the hydrophilic heads here facing the water, hydrophobic tails facing the inside. that they basically form these spheres. Again, because the inside, these nonpolar ends don't like water. Okay, next class of biomolecules. So we learned about carbohydrates. Remember the monomer for carbohydrates, a monosaccharide. And we can take those and form polymers like disaccharides and monosaccharides. Or sorry, disaccharides and polysaccharides are your polymers. Let's talk about proteins. These are the most abundant and most important organic molecules. They are much of the activity that happens in your cells and in your organs is based on the activity of proteins. All right. Different elements in them: carbon, hydrogen, oxygen, nitrogen, right? We've learned about those, very important elements. So there's different monomers. So just like for carbohydrates, the monomers for monosaccharides, the monomers for proteins are amino acids, and there's 20 of them. And you're going to have different combinations of those that are going to form your different proteins. Those are your polymers. Okay, pretty important slide here. So what do proteins do? Okay, so I could see a fill in the blank question asking you, what are 2 or 3 functions of proteins? And they're right here. Okay, so this is something you need to make sure you know. All right, so they can provide support in the case of structural proteins. They can be involved in movement in the case of contractile proteins. They can be involved in transport in the case of carrier proteins or other transport proteins. They can be involved in buffering and help regulating pH. They can be involved in metabolic regulation, which is this basic controlling chemical reactions. And enzymes do that. Okay, I think we learned a little bit about enzymes lowering the activation energy already. Proteins are involved in coordination and control through hormones. So some of your hormones are proteins. They can control responses in your body. Proteins can also be involved in defending your body against invaders through antibodies, okay, which are proteins. So, and I wouldn't necessarily ask you to give me lots and lots and lots of information about that, but I mean, you should be able to say support, movement, transport, if I asked you for 3 of them, right? Or whichever ones that you remember. But you don't know which ones I'm going to ask you. And the other thing is, it could also show up as a multiple choice question. It could show up as a matching, so I wouldn't say just know 2 or 3 of them. Know as many of them as you can remember, because it could show up in different ways. I'm just trying to give you examples of how it could show up as a question. All right, so protein structure. Proteins are composed of long chains of amino acids. We talked about that. Amino acids are the monomers. Each amino acid has certain building blocks. You have to have a central carbon atom, okay, that holds everything together. Well, also, that carbon is going to have basically 4 attachment sites, right, because it can form 4 covalent bonds. We'll have a hydrogen atom, an amino group, which is NH2, a carboxyl group, which we learned is COOH, and a variable side chain or R group. So that's what changes. Okay, the other 2 through 4, or really 1 through 4, are going to be the same for every amino acid. What's different will be number 5. All right, so here's what that looks like. Here's your central carbon atom. Here's your amino group, your NH2. your hydrogen atom, your carboxyl group, and your R group, your variable side chain. Okay. This is what changes. Okay, linking amino acids together, creating polymers, requires what process? We learned about this with carbohydrates. What is it? Dehydration synthesis. Okay, same process, just different biomolecule. So you're taking the amino group from one amino acid and the carboxyl group from another amino acid and creating what we call a peptide bond. The molecule that's created is a polymer, which we call a peptide. And a polypeptide is anything that's 3 peptides or larger. Okay, so anything has essentially 3 amino acids to it or more. All right, so here's forming the peptide bond. There's 2 amino acids. Okay, I'm not asking you to memorize all of them. What I am asking you just to know this is what changes. Okay, the side group. Okay, take the OH from the carboxyl group, hydrogen from the amino group through dehydration synthesis. We're synthesizing this new peptide and you're releasing water, so dehydration synthesis. Now, if you do the reverse reaction, you use water, break the peptide bond, and you get the amino acids again. Okay. So hopefully you're starting to see what dehydration synthesis and hydrolysis mean. Okay. You see it over and over again. All right, so how do we describe the shapes of proteins? Well, proteins have different levels of structure. The primary structure is just your amino acid sequence, just the exact amino acid sequence as it is. The secondary structure is if you take into account hydrogen bonds. So hydrogen bonds are going to form spirals or pleats, and we'll see what that looks like in just a minute. But it's all based on folding based on the hydrogen bonds in the polypeptide itself. Tertiary, anybody know what tertiary means? Yeah, so tertiary is the word for 3. So 3-dimensional shape. Okay? So we're not just looking at the hydrogen bonds, we're looking about how the whole molecule coils and folds to produce a 3-dimensional shape. So you might have a secondary structure in one part of the molecule, but we're looking at how the whole molecule folds. Then you have quaternary structure. Quaternary is the sort of 4th level or higher level, and it has to do with how multiple polypeptide chains interact with each other. So primary, secondary, tertiary just deal with one chain. Quaternary incorporates the fact that you could have multiple chains connected. All right, here's our primary structure, our chain of amino acids. Here's our secondary structure, takes into account, in this case, spirals, because you've got hydrogen bonds that are allowing these interactions to happen on the molecule. And again, I don't necessarily need you to figure out, like, well, how do I know where the hydrogen bonds are? Or how do I know where they fold? Or how do I know where they do a spiral? You don't need to know that right now, okay? What you do need to know is secondary structure takes into account hydrogen bonding. And the 2 examples of secondary structures are alpha helices and beta pleated sheets, okay? I think if you want to take, a full semester of biology and a full semester of biochemistry, we can, get into some of that for you. But my guess is most of you are here because you really want to be nurses or physical therapy assistants and you want to get on to doing that stuff, right? So we can teach that to you at some point. But what I don't want you to do is be like, well, I don't understand how this works, so therefore I just check out. You don't have to know everything. Okay, I'm trying to make it where I'm telling you exactly like this is what you need to know. Don't worry about the rest of it. Okay. All right, so primary structure, so we covered that. So here's the other example of secondary structure. Here's your beta pleated sheet. So it just means depending on the arrangement of the atoms, you might get what looks like folds, or you might get what looks like spirals. Okay, and that's all based on hydrogen bonding. Here's your tertiary structure. So it's looking at the whole polypeptide. You can see some coils. All right, but it's basically how the whole molecule moves, what it looks like in 3 dimensions. Quaternary structure, multiple subunits, multiple protein units together. That's important because anybody heard of hemoglobin? Okay. Hemoglobin is used in your bloodstream to carry oxygen to your body, right? So hemoglobin is most effective when it has 4 subunits and allows it to carry more oxygen. Okay, so you want to be able to carry as much oxygen to your body as possible, and so it works. You wouldn't just want 1 subunit at a time. You actually want it to be clumped together as 4 subunits of hemoglobin working together to carry more oxygen. So that's the difference between quaternary and tertiary structure. All right. Here's another example. For collagen, you've got multiple subunits that wrap on, so each one has its own spirals, but then the whole subunit starts spiraling on each other. So that's just another example of quaternary structure. And you want that because it makes this fiber very strong, and there are parts of your body where you need those strong fibers. Okay. You also have introducing with, when we just talked about collagen, you have fibrous proteins and globular proteins. All right. So fibrous proteins, basically sheets or strands, and their shape is determined based on their secondary structure or the quaternary structure. Okay, it's based on the hydrogen bonding, or it's based on multiple subunits. That's another way of saying that. All right. Globular proteins are basically spheres. They're round, and they're soluble, which means they dissolve, and they have some function in your body. But their shape is determined based on their tertiary or 3-dimensional shape. All right, so enzymes. So enzymes are catalysts. Okay, so we said that was a property of proteins, that they can serve as enzymes. So what do catalysts do? Well, they lower the activation energy in a chemical reaction. So the way I would describe that is, if, let's just say, maybe it's your goal at some point in your life, you want to be able to buy your own home. And let's just say you are working in this program so that you can do that. You finish your program and you go to the bank and you say, I want to buy a home and I need to take out a loan. And they say, well, what do you do? Because we need to know if you're a good risk or not. And they say, well, I'm a nurse, or I'm an occupational therapy assistant. Oh, well, guess what? Since you're a healthcare provider, instead of having to provide the full down payment, you get a reduced down payment because you're in this profession that we think is great. So the bank just lowered your activation energy. They said, well, instead of you having to have this much money, you only have to have this much money. Okay? That's basically how enzymes work. Also, in a sense, the other piece of that is that the enzyme gets to use these over and over again. So it would be like there's this magic pot of money, and you got to use it, and then, you know, basically you start paying that money back, and then they give that money to somebody else, and then another nurse can use it, another occupational therapy assistant can use it. So it's not used up. All right. Another piece of enzymes is that enzymes have what we call an active site on them. So basically, if you have this clump, which is your enzyme, there's a little spot on the enzyme where a substrate or a reactant can attach to it. Once it does that, then the chemical reaction happens. All right, different properties. You also see enzymes will show or exhibit specificity. So usually they'll only catalyze one type of reaction. Okay, they're very specific. They have what we call saturation limits. So an enzyme can only take so much substrate. So it'll lower the energy activation, but it's kind of like saying, I only got so many down payment credits to give out. Like once I use them all up, I can't give any more out, so I get some of my money back. Okay. All right. Regulation. So sometimes enzymes are regulated by other chemicals. Okay. Sometimes they can't just do whatever they want to do. They have to get essentially other chemicals in place before they can act. All right, so here's a picture of that enzyme substrate complex. So first step, here's your enzyme. It has an active site. You can see it's very specific because the attachment sites have to match. So your substrates, your reactants, basically have to interact and meet up at the active site. So once the substrates attach to the active site, you form this complex, and that makes the reaction able to happen. All right, so the substrate binds and it changes shape. So that basically once the substrate binds to the enzyme changes shape, and in so doing, it creates a reaction that creates this new product. You're taking two monomers and making a polymer. Once that polymer is made or that product is made, it's ready to be detached, and it leaves the enzyme. The enzyme's ready to go again. Okay. All right, cofactors. So what are they and how do they relate to enzymes? Well, so cofactors are an ion or molecule that binds to that enzyme before the substrates can bind. Remember we said that sometimes there's regulation? So this would be an example of that regulation. Okay, and you also have coenzymes. And these are cofactors that are not proteins. Okay, so typically enzymes are proteins. In this case, a coenzyme is not a protein, but it has a similar effect. And vitamins are an example. Okay, temperature and pH. How do they affect enzymes? Well, here's a term you need to know, denaturation. And this denaturation just means the protein changed shape and lost its function. Why? Because it got too hot, or the pH went out of the range that it will work in. Got too acidic or too basic. Okay, so it became denatured. Doesn't work anymore, change shape. All right. Other types of proteins, you have glycoproteins. These are very large proteins, and they have small carbohydrates on them. And the way that I remember these is like the part at the beginning, these are sort of adjectives, which means that they sort of modify the noun. So protein is the noun. So the protein is large with small carbohydrates on it. And it has enzymes, or enzymes can be glycoproteins, antibodies can be glycoproteins, hormones can be glycoproteins, parts of the cell membranes can be glycoproteins. Mucus contains glycoproteins. You also have proteoglycan. In this case, glycan is the noun, proteo is the adjective. So this is a large polysaccharide with some polypeptide on it, or some protein on it. So their function is to increase fluid viscosity, which means our fluid thickness. So there are times when you want fluids in your body to be watery and times when you want them to be thicker. And proteoglycans are involved in that. Okay, so let's finish up with nucleic acids. So these are large organic molecules, and you'll see them in the nucleus of your cells. What are they for? For information storage and processing. Your classic examples of nucleic acids are DNA and RNA. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. Okay? It's what determines your inherited characteristics. Okay? And it's what controls synthesis of proteins. Also controls production of enzymes, which in most cases are proteins, right? It also controls metabolism, which is basically the chemical reactions that happen in your body. So pretty important. Another nucleic acid is ribonucleic acid, or RNA, and it controls the intermediate steps in protein synthesis. Okay, so we'll learn more about the relationship between DNA, RNA, and protein, but ribonucleic acid is what's involved in the intermediate steps. All right, so structure of nucleic acids. Okay, long chains of nucleotide monomers. And each nucleotide is going to have these things. It's going to have pentose sugar. Pentose means 5-membered ring. Pentose sugar. With deoxyribose or ribose as a sugar. If it has deoxyribose, it's DNA. If it has ribose as a sugar, it's RNA. They'll all have a phosphate group, and then they'll have nitrogenous bases in each. So one nitrogenous base per nucleotide. And those are A, G, C, T, and U. Okay, and we'll find out which ones go where in just a moment. Here's your structure. All right, so you have the sugar, pentose sugar, phosphate group, nitrogenous base. And the nitrogenous base can be what we call a purine or a pyrimidine. Okay. So your purines are adenine and guanine. There's two ways you can kind of remember that. One is pu and purine. Pu rhymes with two because they have two rings. Another way I used to remember because I went to Texas A&M and we were the Aggies, and we say Aggs are pure. And so it's whichever way makes sense to you. All right, so adenine and guanine are the purines. Cytosine, thymine, and uracil are the pyrimidines, and they only have one ring. You'll only see thymine in DNA, and you'll only see uracil in RNA. Okay, so that's an important difference between the two. Thymine is in DNA, uracil is in RNA. All right, so DNA has two chains, two nucleotide chains, two strands. RNA has one strand, so that's another difference. DNA has two strands, or one chain of nucleotides. RNA has one chain of nucleotides. Okay? DNA is going to have hydrogen bonds between the nitrogenous bases of the different strands, which hold the two strands together. So if this is a strand, this is a strand, those nitrogenous bases are on the edges and they hydrogen bond with each other. And it forms a twisting double helix, so it spins on itself. All right, 3 different types of RNA: messenger RNA or mRNA, transfer RNA or tRNA, ribosomal RNA or rRNA. Let's see. Okay, all right. So, like I said before, you have the base pairs and the different strands, and they have to be complementary to each other. So purines will base pair with pyrimidines. So a purine on one strand will pair with the pyrimidine on the other strand. Adenine bonds with thymine. Cytosine bonds with guanine. This is for DNA. Okay. Now, the only wrinkle here is that, for RNA, adenine still, basically, uracil is going to replace thymine. So cytosine and guanine will still bond to each other. But there's no thymine in RNA. So if there is a adenine in one strand and you're making RNA, the other strand is going to have uracil. Okay, uracil replaces thymine in RNA. All right, so here's your single-stranded RNA molecule. Has uracil in it. No thymine. Here's your DNA strand. Okay, you can see your nitrogenous bases on the inside of the strands, connected by hydrogen bonds. A binds with T, C binds with G. All right, here's the overview. Definitely want to make sure you look back at this in the canvas. Nitrogenous bases, RNA is A, C, G, and U. Nitrogenous bases for DNA, A, G, C, and T. Sugar for RNA is ribose. Sugar for DNA is deoxyribose. All right, number of nucleotides in a typical molecule varies from under 100 to 50,000. Number of nucleotides in DNA is 45 million or more. Shape of the molecule varies with hydrogen bonding along the length of the strand, right? Whereas DNA is always in a double helix. Function: RNA performs protein synthesis under the direction of the DNA. DNA stores the information that controls it stores the information that controls protein synthesis. So DNA is directing RNA. Okay, give me a couple minutes and we'll finish this chapter, start chapter 3 next time. So your high energy compounds are derived from nucleotides. The process of taking and adding a phosphate group to another molecule is called phosphorylation. This is adding a phosphate group. And that bond that's made has very high energy. Examples: you have a nucleotide, an adenine with one phosphate, or adenosine with one phosphate, it's adenosine monophosphate. You have 2 phosphate groups, adenosine diphosphate, and adenosine triphosphate is your high energy compound with 3 phosphate groups in it. And that's what we'll spend a lot of time talking about in this course. Adenosine triphosphatase is an enzyme that catalyzes conversion of ATP to ADP. So you're removing a phosphate. All right, so anytime you see ASE, that's an enzyme. Okay, that's an enzyme. ASE is enzyme. All right, so we'll finish with this. So you have your high energy bonds. Again, here's your adenine and ribose, forms adenosine. Add a phosphate, adenosine monophosphate. Adenosine diphosphate has 2 phosphate groups. Adenosine triphosphate has 3 phosphate groups. Okay. So Okay, what I need for you to do now is to work on your exit tickets. Okay, you can do that on your phones or your computers.